we're going to start with something that I hope is mostly review for you guys, which is DNA replication. Okay? So, hopefully you're all familiar with the basics of this. You know the bases. You know A pairs with T, G pairs with C, or A can pair with, yeah, you don't even need to know about U yet. And the fundamental reason to have DNA replication in the cell is to duplicate the cell's genome. That's the single most important reason for having DNA replication. And hopefully you all know that the genome basically contains the blueprint for every cellular function. Okay? I don't want to get into whether DNA determines every aspect of us. It's pretty sure that's not. But certainly all fundamental events of a cell are controlled by the cell's genome. So obviously you want this process to work well and to be very accurate. Okay? The other thing about the replication process is if you do make mistakes, there's often consequences. There are potentially good consequences. You can develop a new attribute that is good for your survival, okay? And this happens with some frequency. But more often than not, there are bad consequences, okay? And that can be anything from straight-up lethality, okay? The organism can no longer grow, to things like cancer or a new attribute that actually makes it so an organism less able to grow. Okay? And because more generally the outcome is the latter than the former, again, you'd like this to be as accurate as possible. Okay? And we'll talk about how it becomes accurate as we go along. Okay, so what are the properties of DNA replication? First of all, it's rapid. Okay? It's somewhere between 100 to 1,000 base pairs per second are synthesized by a typical DNA replication machine. Okay? It's also complete for the reasons that we just talked about. So essentially, and there are a few exceptions to this, oops, every base pair must be replicated in the genome. Okay? We'll talk about telomere replication at some point. That's one example where not every base pair is replicated. And the final thing is it's extremely accurate. Typically, the replication machinery makes one mistake in every 10 to the 10th base pairs synthesized. Okay? There's two ways to think about this. You can think about it in terms of E. coli. So E. coli has a genome of about 5 times 10 to the 6 base pairs. And that means it's going to make a mistake about each organism, one out of every roughly 1,000 organisms will have a mistake in its genome. Okay, so that sounds pretty good, actually. I like those numbers. It's a little more scary if you're a human, right? So we have a genome of 3 times 10 to the 9th. So that means we make a mistake about 1 every 3 cell divisions. Okay? But it's worth keeping in mind, most of those 10 to the 10th base pairs are not coding and are not that important to be correct. But if it's in the middle of a coding region, it changes the amino acid in a protein, it can be a problem. Okay? So we're pretty much full of errors, is one way to think about it. Even under the best of circumstances, we have thousands and thousands of errors. Okay? I'll tell you one other fun fact about DNA replication. Who knows how much DNA is in each one of your cells? How long is it? You think it's 2 inches? 2 meters. Okay? How much in your whole body? It's enough to go to the sun. Okay? It's about 90 million miles. Okay? How much do you synthesize in your lifetime? It's a light year. Okay? So it's pretty impressive machinery that can do that and not end up with us being sort of a bubbling mass of mutant cells that just sort of sits around like a pile of jello. Okay? So this is an incredibly important, very accurate process that makes sure that we stay who we are throughout our lifetimes. Okay, so basics. let's talk about, oh, actually, one other thing. The studies of replication led to some very important things that all of you probably know about. So molecular cloning, the fact that we can splice genes together or make all sorts of different things, directly came from studies of DNA replication. Okay? PCR directly came from that. I have a third thing there. Oh, all of this massive sequencing. Okay? The fact that we can sequence entire genomes, including our own, is a direct result of studying DNA replication. And then the other side of it is that DNA replication is very frequently the target of drugs. So most frontline chemotherapies target DNA replication. Okay? There's lots of fancy new ones that lots of people are developing and hopefully will work much better than the DNA replication ones because unfortunately they typically not only make your cancer cells sick, but they make all your other cells sick. But if you get cancer, the odds of you not being treated with a DNA replication inhibitor is extremely small because most of these fancy new things are used either in collaboration with those or are not used for the particular type of tumor you have because they mostly are targeting very small numbers of tumors that can be gotten. And the other thing is most antivirals target DNA replication. So for example, there is one of the best anti-AIDS drug targets the DNA replication of its genome. Okay, or RNA/DNA replication of its genome. So these are very important targets, and so understanding replication helps us to hopefully develop new targets, and if nothing else, maybe make some that won't make us quite so sick when we treat them.